Hello, my name is Kate Musto, and this presentation is going to be on the regulation of the gastric phase of digestion. I included this picture diagram of the stomach, and this is what the stomach looks like when there's no food and it is empty in the fasting state. Accommodation is when food is going to enter the stomach, and as you can see here, there are stretch receptors in the stomach that we will discuss later. And then the emptying of the stomach is when food is going to be eliminated into the small intestine. So the gastric phase occurs in the stomach after the cephalic phase. It is a series of short reflexes, and if you look over to the diagram on the right, you can see that there's the vagus nerve, which is cranial nerve 10, which is often associated with the parasympathetic nervous system in aiding, digestion and smooth muscle contraction and relaxation. So as you can see, when food enters the stomach as a bolus, stretch receptors in the simple stomach area towards the top of the fundus are activated. And then there is a cascade that comes down through the stomach. This is the bolus right here. And the parietal cell, which is also going to be attacking the bolus in order to start digestion. And then we'll go into detail further of what the parietal cell and the gastric stem parietal cell, the G cell, and everything is used for later in the lecture. The stomach is lined with gastric pits and glands responsible for producing acid, hormones, paracrine molecules, and enzymes. And there are two subcategories, one being storage digestion in the stomach, which is chemical and mechanical digestion, into chyme, which is more of a liquid substance, and then the defense of the stomach, which protects us from absorbing any pathogens or bacteria. I included this slide because it's a flow chart that includes everything for gastric function. In the first stage, the food or cephalic reflexes initiate gastric secretion of gastrin, histamine, and acid. We'll go into those further later in the lecture. Also, the gastric phase is initiated after the cephalic phase, and in the gastric phase is where 2/3 of the acid and the hormones are produced in order to aid in digestion. Gastrin stimulates acid secretion by direct action on parietal cells or indirectly through histamine. So we're at this portion of the diagram here. The acid stimulates short reflex secretion of pepsinogen. Now, pepsinogen is the inactive form of the enzyme pepsin, which discuss soon. And the somatostatin released by hydrogen is the negative feedback signal that modulates acid and pepsin release. Next slide. So we're going to start with acid secretion and digestion. The parietal cells from the gastric gland secrete gastric acid into the lumen of the stomach, which makes the stomach very acidic. It can be as low as pH 1, but is typically around pH 2 to 3. The functions of gastric acid include denaturation of proteins via breaking disulfide and hydrogen bonds in the amino acid sequence of a protein. It helps kill bacteria and other ingested microorganisms. So as I was saying before, that's more of the protection of the stomach. And then it inactivates salivary amylase and stopping carbohydrate digestion. So as you can see on the diagram below, when the food enters the stomach, the stretch receptors send the signal to the brain, which triggers secretion of digestive juices. The stomach does release gastrin, which promotes the production of stomach acids, such as the gastric juice and then the acids that are released from the parietal gland. And then, just as an important note, the intestine does release secretin and cholecystokinin, which is the stimulation secretion of pancreatic juice and bile, which is just aiding the stomach in the gastric phase in order to digest different substances. Now, for enzyme secretion and digestion, there are two main enzymes that we're going to focus on for today. The first being pepsin, which is the initial digestion of proteins. Pepsin is secreted by chief cells, and like I was mentioning before, the inactive form is pepsinogen in the gastric glands, which then is activated to form the active pepsin, and this is particularly beneficial in protein digestion. And then secondly, the gastric lipase is more geared towards the breakdown of triglycerides. It is co-secreted with pepsin because they're most likely, when there's food or anything coming into the stomach, it's not just going to be one substance, so multiple enzymes have to be elicited in order to do proper digestion and not have anything rejected from the stomach. And then only a small portion of fat does occur in the stomach. The remainder of the fat absorption and digestion does happen in the small intestine. So if you look at the diagram on the right-hand side, here again, it just reinforces the idea of distension with the stretch receptors and the pH being elevated from the gastric juices that we had mentioned before. And then there's a feedback mechanism with chemoreceptors coming down to the myenteric plexus. And then we have all of our cells and what they're secreting in the stomach. Mucus cells doing mucus, which is also helping to protect us against any ulcers. Chief cells secreting pepsinogen, and parietal cells secreting the gastric juice hydrochloric acid. Now we're going on to paracrine secretion of digestion. This is including our histamine, somatostatin, and intrinsic factor responses. So our histamine, first of all, is a paracrine signal secreted by the ECL in response to gastrin or acetylcholine. Now, if you look quickly over to the right-hand side of the diagram, we have our ECL cell right here, and the neuron receiving any input, which is going to activate the parietal cells. And then again, our G cells, that's going to help aid in circulation of the absorbed nutrients from the bolus and the chyme that was digested in the gastric phase in the beginning. And then next, our somatostatin is a hypothalamic growth hormone inhibiting hormone secreted by D cells in the stomach. This is our primary negative feedback mechanism for our gastric secretion. It does shut down the acid secretion, and it inhibits pepsinogen secretion from the chief cells. This is so we're not doing over-digesting and we end up hurting the mucosa of our stomach. And then the intrinsic factor is a protein secreted by parietal cells, specifically for B12 absorption, necessary for red blood cell formation. If we lack any one of these, paracrine or even the gastric juices or the enzymes, digestion in the gastric phase would not be as efficient. An example of that is the intrinsic factor. If we lacked intrinsic factor, it could lead to pathology such as pernicious anemia, and we're unable to form red blood cells. So that was our lecture for the regulation of the gastric phase of digestion. The next one will be about the reproductive system.